Welcome, Dr. Bishal Chhetri. It's my pleasure to introduce the speaker for today's talk, Dr. Bishal Chhetri. Dr. Bishal Chhetri is currently working as an postdoctoral fellow at the Department of Mathematics and Statistics, IIT Kanpur. Before joining IIT Kanpur, Dr. Bishal worked as a research scientist at Karkinos Healthcare Private Limited, where he worked with the leading scientists and the oncologists in the field of early cancer detection. Dr. Bishal completed his bachelor's, master's, and PhD from Chhetri Institute of Higher Learning. During his PhD, Dr. Bishal worked on within host, between host, and multi-scale dynamics of COVID-19 and optimal control problems. Dr. Bishal expertise is in the areas of disease modeling, mathematical oncology, and nonlinear dynamics. His current research focuses on developing stochastic mathematical models to gain insights into cancer progression and building machine learning and deep learning models for the early detection and classification of cancer. Dr. Bishal's research and findings have been published in prestigious journals. He actively shares his knowledge through talks and presentations at workshops and conferences. On the personal front, I have taught Dr. Bishal at the undergraduate level at Brindavan campus in Chhetri Institute of Higher Learning, and he has been a very hardworking student, and we can see the accomplishments what he has achieved today because of his hard work and perseverance. So, with these few words, now I welcome Dr. Bishal to talk on the topic understanding cancer disease using mathematical and machine learning models. Over to Dr. Bishal.
Thank you so much, sir, for the introduction. So let me share my screen. Is my screen visible, sir?
Yes.
Okay. Okay, so I hope I am also audible.
Yes, yeah, you can go ahead.
Okay, yeah, sure. Thank you so much, Darshan sir, for the introduction. And loving Sairam to all of you present here in this talk. And before starting my talk, I would like to invoke Bhagwan's divine presence and his blessing. So, in today's talk, I thought I'll speak on cancer disease and different mathematical models that are developed for understanding the progression of cancer disease. So I'll be starting with what is cancer disease in general, the different biological aspects of cancer disease, the various challenges, and then I'll build a motivation for why do we need to build mathematical models to understand cancer disease. In this case also, I'll speak from the exponential model to stochastic differential equation models that are developed for understanding cancer dynamics. And then in the second part of my talk, I'll talk about the machine learning models that we have used for breast cancer prediction. And then I'll be concluding my talk with the current ongoing research works that we are currently doing. So for better internet stability, I would like to switch off my camera so that there is no disturbance in the in between the presentation. So let me start with general definition of cancer. So what do we mean by cancer? So cancer today is understood as a disease. It is not just a single disease, but it is a group of diseases that is characterized by the uncontrolled growth and spread of abnormal cells. So from this, we see that cancer is not a particular type of disease, because there are several types of cancer based on their origin. And when cell, when the cell in the body becomes abnormal, it starts growing, uncontrolled growth of cell happens. And in case of cancer, it also spreads to the various parts of the body, causing several damage to different organs of the body. So therefore, cancer is called a group of disease, and it is not just a single disease. Now, let us understand how this understanding of cancer happened over the period of time. In around 1400 BC, there was what is called Hippocrates theory, that was widely accepted at that point in time. And this theory described cancer as a growth or mass that is caused by an imbalance of blood or bile in the body. So this was the understanding in and around 400 BC. And Hippocrates is also credited for originating the term cancer and carcinomas. So he was the first person to use the term carcinomas for ulcer causing tumors. So later, the study increased in cancer, and in 1600 and 1800s, there came some pioneering studies which said that tissues are composed of cells. So with this study, people started thinking cancer as composition of cells, because tumor is also a tissue. So with this, researchers started researching on how these cancer cells could be different from the normal cells, and what could be the driving force behind the cell behaving abnormally. In the same time, in the mid of 1800s, there were some studies that revealed that the person who are cigarette smokers and those who work in the mines, they have a higher chances, they are at least 10 times more likely to develop uh, a lung cancer than the non-smokers. So this kind of studies were done in the mid of 1800s. And from this, people also started believing that cancer could be caused because of some external factors, and that can also be avoided. So in the case of lung cancer, if smoking is avoided, then they felt that lung cancer can be prevented. So these were the general understanding of cancer disease. And today, with the evolution of science and technology and the current understanding of molecular biology, today cancer is said to be caused because of the genetic and epigenetic alterations or modifications. So what are these genetic and epigenetic modifications or mutations? So at the DNA level, if we see, a gene is nothing but it is a sequence of nucleotides, ATGC, that we know, right? And when gene gets altered, what exactly happens? So gene is actually, it is, it contains an information for producing protein, and this protein has various functions. They regulate various functions in the body, from the cell growth, division, repair, and also death. So when a gene gets mutated, or when a expression of gene gets overexpressed, in that scenario, the protein that is supposed to form gets disturbed, or it gets perturbed, and the particular function that it is supposed to do, it will not do that particular function. So in that case, if we see a protein responsible for, let's say, apoptosis, so if that protein gets perturbed or disturbed, then it will not perform the function of apoptosis. So there is no cell death happening, and therefore there is only growth of cell, and there is growth of cell, and that causes cancer. So this is a way of kind of understanding the cause of cancer. Now, what exactly differentiates, or what are the fundamental characteristics of cancer cell from normal cell? So what are the features based on which we can say this particular cell is cancer and it has a distinguished feature from normal cell? So there was a pioneering study done in the year 2000, and the work of the title of that particular work was The Hallmark of Cancer by Weinberg and his team. And this paper is considered to be one of the main paper in understanding cancer disease through various characteristic features. So here what they said is, they said that cancer disease could be understood through a set of underlying principles, and this set of underlying principles are common across most of the cancer types. So most of the cancer types share common characteristic feature that helps them to distinguish from the normal cell. And this characteristic feature is what is called the cancer hallmarks. There are 6 cancer hallmarks that were presented in this paper. The first being the self-sufficiency in the growth signals. The second is the insensitivity to anti-growth signals. So in this case, the tumor cell becomes insensitive to anti-growth signals, and therefore there is a growth or proliferation of cancer cell. They also evade what is called apoptosis, the programmed cell death. There is a limitless replicative potential of tumor cell and sustained angiogenesis that helps in the growth of tumor cell. And the last hallmark is tissue invasion and metastasis. So these were the 6 hallmarks that were presented in this paper, that is the hallmark of cancer, published in 2000. And based on this characteristic feature, the authors in this paper said that cancer could be understood using these features. So in the next couple of slides, I'll be talking a little more of biology of cancer, where I'll be discussing each of these hallmarks, because for mathematical modeler, like we need to kind of have a understanding of each of these pathways to come up with a mathematical model to represent this system in terms of equations. So it is very important for us to kind of have a understanding of this characteristic feature so that we can come up with a efficient mathematical model. So let us see the first cancer hallmark, that is self-sufficiency of growth signals. Now, for a normal cell to divide, it relies on the signals from the neighboring environment. So when a cell has to divide, it has to get some signals from its neighboring environment, and based on the signals, so there could be like anti-proliferative signal, or there could be proliferative signal. So the cell weighs the signal, and based on this, like if anti-proliferative signal remains stronger, then cell will not commit to go for cell division. Whereas if proliferative cell signal is high, then uh, they start the process of cell division. So this is what happens in a normal cell, in a normal cell scenario. Whereas in case of tumor cells, they show reduced dependence on external growth stimulation. So they are found to acquire their ability to synthesize their own growth factors. So this happens through multiple ways. And one way is where these tumor cells are found to produce what is called PDGF, platelet derived growth factor, and TGF alpha. And this enables the cancer cell to become self-sufficient, and they no longer depend on some external signals for cell division to happen. And therefore, using these growth factors, they keep sending signal to the nucleus, and then cells keep dividing. Another way of understanding this is where, like, the cancer cell uses what is called oncogenes. So normally what happens is there are some genes that are responsible for growth and division. And when these genes, this type of genes that is responsible for cell growth and division, they are what is called proto-oncogenes. And when these genes get mutated, in that case, what happens is, in that case, they produce their own growth signals, and therefore they do not depend on any external signals for cell division to happen. So these are the ways in which cancer cell become self-sufficient in sending the growth signal for cell division to happen, and then the cell division keeps happening uncontrollably. The second cancer hallmark is insensitivity to anti-growth signals. So in human body has a mechanism, a counter mechanism. So if something is high, so there is a mechanism to kind of maintain an equilibrium of the system. So in this case, like to counter the oncogenes are genes that help cancer cell to divide by having their own growth signals. So to counter this mechanism, there are what is called tumor suppressor genes. And some of the examples of these genes are P53 and P16. But cancer cell, again, it tries to inhibit this tumor suppressor gene, and they escape from this tumor suppressor gene also. So how do they do that? So cancer cell, what they do is they release what is called TGF beta. This is a cytokine, and with this, they inhibit the function of tumor suppressor genes. So by releasing this cytokine, they suppress the activity of tumor suppressor gene, and therefore there is no tumor suppression happening, and they keep kind of they create an environment where they can grow and proliferate. So with this, cancer cell grows and proliferates, switching off the tumor suppressor genes. So this is another pathway in which cancer cell keep dividing and proliferating. So this figure depicts a case where there is a tumor suppressor gene, and this is how it looks in the normal case. And if suppose it gets mutated, say for an example, because of some external factors like smoking or radiation, this function of these genes get altered, and this tumor suppressor genes, they no longer function, and because of this, there is a accumulation or proliferation of cancer cells. So this is a second hallmark. So from this, we kind of see that there is a production, there is a dynamic change in the production of this kind of molecules here, cytokine levels, and there is a interaction between different signals, different cells in a and this interaction kind of based on this interactions, tumor either grow or they proliferate, or they grow or they do not grow. So that actually depends on the dynamic interactions between these cytokines and different cell types. So therefore, this kind of systems can be represented by dynamical systems that are nothing but mathematical models. The third type of cancer hallmark is resistance to cell death, that is apoptosis. Apoptosis is nothing but programmed cell death. Whenever a body feels that there is a damage or there is unwanted or harmful cell in the body, it gets removed through the process of apoptosis. And in the case of cancer, tumor cell, they are found to stimulate and activate anti-apoptotic pathways that does not allow the cancer cell to die. So there are some pro-apoptotic proteins such as BAX and BAK, and tumor cells, what they again do is they are found to alter the function of this pro-apoptotic proteins. Therefore, if these proteins whose function is to drive the process of apoptosis, if they get mutated, then their function is altered, and therefore they do not do their function of apoptosis. And in that case, there is a growth in the tumor cell population. They are also found to enhance the anti-apoptotic proteins like BCL2 and BCLXL. So these are proteins that does not allow apoptosis to happen. So cancer cells, through some mechanism, they are found to enhance the activity of these anti-apoptotic proteins. There is another gene which is called the guardian of the genome, that is P53 gene. It, it has two different functions. It acts as a tumor suppressor gene, and also it acts as pro-apoptotic gene. That is, it stimulates and enhances the apoptotic pathway. But when this gene gets mutated, so when this gene gets mutated or altered, cancer cell, they can evade the process of apoptosis. So they no longer go through the process of apoptosis, and there is no death of cell happening. So this is a third hallmark of cancer, using which they kind of continue to grow and proliferate. Now coming to the fourth one. So there is an interesting fact about uh, cancer cells, where they create an environment to kind of grow limitless. And this is called a limitless replicative potential of cancer cells. And this is given a name, telomere story. So telomere is nothing but it is a sequence of nucleotides. And the function of this telomere sequence of nucleotide is to maintain the stability of the chromosome. So in case of normal cell, what actually happens is, like, normal cell, when they go through the process of cell division, after few division, the DNA strand become unstable, and then there is a process of apoptosis. So normal cell cannot divide infinitely. Whereas in the case of tumor cell, what it is found is, and there's one more thing that I want to kind of tell here. So there is this telomere. They are actually located at the ends of the DNA strand, and each time the DNA gets replicated, the end of the telomerase, it gets cut and becomes shorter and shorter. So the DNA strand is there, and the end of the DNA strand is where telomere is located. So when cell division happens, the telomere gets shorter and shorter, and after multiple cell division, this telomere becomes too short that it creates instability in the DNA strand, and then there is a process of apoptosis happening. So this is what happens in a normal scenario. Whereas in case of now, if we see in case of cancer cells, the cancer cell is found to upregulate the expression of what is called telomerase enzyme. So there is one enzyme called telomerase, and using this enzyme, they succeed in maintaining the length of the telomere. So they kind of uh, regulate the expression of this telomerase, the enzyme, and then they are successful in kind of maintaining the length of the telomerase. And therefore, if the length of the telomerase is maintained, the DNA strand is, there is no instability in the DNA strand, and there is a kind of unlimited multiplication of cell that keeps happening. So this is another way in which there is limitless replicative replication of cancer cells. Now coming to the last cancer hallmark, this is the hallmark using which cancer cell, encourages the growth of capillaries. If we see, for any cell to grow, it requires oxygen supply and it requires nutrients. So is the case with respect to cancer cell also. So cancer cell also needs nutrients and oxygen to grow large, and this happens through the process called angiogenesis. So angiogenesis is nothing but a process where formation of new blood vessel happens in the tumor cell. And there is another interesting thing. So cancer cell also is found to trick the body by making the body understand that cancerous mass is nothing but a wound. So when a body understands that cancer mass is wound, it uses the repair mechanisms and it supplies food and oxygen for the repair of the wound, thinking that the cancer mass is a wound. And in this way also cancer cell grows. Cancer cells are also found to secrete what is called vascular endothelial growth factors and TGF beta. And these factors, they help in the process of new blood vessel formation through which cancer cell receive the food and the nutrient required for the growth. And with this, like there is what is called angiogenic switch that happens in case of cancer cell. That is, uh, cancer cell remains dormant in pre-angiogenic state. That is, they do not grow, but as the process of angiogenesis starts, they start developing blood vessels, and through that they get all the nutrients and whatever food material is required for their growth. And there is a kind of a switch in the growth of cancer cells. So from the dormant state, they kind of grow unbounded. So this is what is called angiogenic switch. So if we see this cancer hallmark, if we represent this cancer hallmark in terms of mathematical equations, so this we can also understand, like we know mathematical biology, and in mathematical biology we do what is called a stability analysis, where we have a different equilibrium solutions of the system. So this we can understand this angiogenic switch as, like there is a tumor free state where tumor is zero, that is the state. And as there is, as angiogenesis starts, so we can have some parameters that represent the angiogenesis. As angiogenesis starts with that parameter value greater than some threshold, uh, we can show that there is a uncontrolled growth of cancer cell happening. So it reaches to another state, from tumor free state to the state where cancer cancer growth is high. So these were the 6 cancer hallmarks that was presented in the paper Hallmarks of Cancer in 2000. Later in 2011, there were 2 more hallmarks that were added by the same authors. And the title of this work of theirs was Hallmark of Cancer, the Next Generation. So this is the new word that they have added here. So the first hallmark in this was the reprogramming energy metabolism, that talks about the capability of cancer cell to modify or reprogram cellular metabolism to support their growth and proliferation. The second one that they talk about is the cancer cell evading the immune destruction, in particularly the T and B lymphocyte, macrophage, and natural killer cells. So cancer cells, they, through some mechanism, they can also suppress the immune response, and they can even hide themselves from immune destruction. So these were the various cancer hallmarks that I wanted to quickly discuss, because it becomes extremely important for us to understand these pathways, so that we come up with a precise, robust mathematical model. Now, let's talk about the different challenges that we have for understanding the cancer dynamics. So from the hallmarks that we discussed, we find that cancer is not just a disease, it is a group of disease, and it is very, very complex, because there is multiple interaction that happens between various cell types, blood vessels, immune cells, and different other components like the cytokines that we saw. And it is nothing but cancer disease is a multi-scale process. So there is a need for developing a multi-scale model for getting a complete understanding of this. So from this figure, we can see that cancer starts with a genetic mutation. So it starts at the molecular level when gene gets mutated. So when gene gets mutated, there is abnormality in the behavior of the cell, and that reflects in the tissue being damaged, and finally the organ being damaged. So to get a complete understanding of this, it is essential that we combine all of these processes and developed a multi-scale mathematical model. Another major challenge in cancer is the asymptomatic cases in the early stages. So cancer is highly asymptomatic in early stages when symptoms appear, and when patient go to doctor, it would be already cancer would be already in an advanced stage, spread all across the body, and the survival chances becomes very less in this case. So in this case, like cancer detection, early cancer detection becomes extremely important, and this can be done using various machine learning and deep learning models. Now let's come to some mathematics. So we have spoken a little bit of biology, different understood little bit about different pathways that cancer, cancer takes for its growth and proliferation. So how do we kind of bring that biological understanding to equations? So there's a field called what is called mathematical oncology. It is an interdisciplinary field that applies mathematical models and computational techniques for understanding cancer dynamics, predicting, and the treatment, understanding the various treatment in case of cancer. Why do we need mathematical models in cancer? So from the biological aspect, whatever we have seen so far, we have seen that there is a dynamic interactions within what is called the tumor microenvironment that tumor creates. And this dynamic interactions, they influence the growth of cancer, resistance to treatment, and also the interactions within this environment that they create. It also drives cancer to spread to the different parts of the body, that is called metastasis. So we also saw that signaling pathways in cancer, they show highly non-linear behavior such as bistability or switch-like responses that we saw in case of angiogenesis. Like there is a angiogenic switch from dormant to large tumor case. So this kind of bistability is found, and this type of pathways can be represented using mathematical models. Mathematical models also helps designing and optimizing therapies. So when we talk about cancer treatment, the most common treatment that is being used is the chemotherapy and radiation therapy, and this treatment has a lot of side effects. And using mathematical models and the theory of optimal control, optimal treatment therapies can be designed using optimal control therapy theory. So in tumor microenvironment that I was talking, so there are two different environment that gets created when cancer cell interact with different cell types and molecules. So one is where an immunosuppressive microenvironment is created, where using different cytokines like TGF beta, IL-10, VEGF, etc., tumor suppresses the immune response. And another one is anti-tumor immune microenvironment, where the immune cell try to fight the cancer cell and try to remove cancer cell from the body. So this is the motivation for kind of building different mathematical models. So if we go back to literature, there are thousands of mathematical models that are developed using using differential equations, using stochastic differential equation, PD based mathematical models are also there. So in this what I thought is I'll briefly talk about some of the mathematical models that are developed and also highlight the results from this study. Now, like any other study of disease, like exponential model is one of the simplest model that is known. And initially, exponential models were used for understanding the growth of tumor, though they have limitations here, and that limitations were taken into consideration, and the models over the period of time. So this is the exponential model, and the assumption here is there is no growth constraint. So there is no growth constraint. All the nutrients that is required for the growth of tumor are available in abundance. So, and because of all this, like there is a the predicts the unbounded growth of tumor. So this is how the modeling in cancer evolved with the exponential model initially. Now, after this, with several in vitro and in vivo experiments, they found that exponential growth of tumor happens only for a relatively short period of time. As tumor grows over the period of time, there is a competition between the tumor cell for survival, right? So only the fittest cancer cells survive. So there is a, a fighting between the cancer cell for survival, and because of this, uh, the growth gets retarded. There is a retardation that limits the growth over the period of time. And in this case, there is a switch from exponential growth to linear growth. And this kind of scenario is modeled using logistic equation, and that is given here. So in this case, we find that when the tumor population is less, there is a linear or exponential growth, and as the tumor population approaches k, here k is the carrying capacity, as v becomes k, then there is a linear growth. So this kind of gives us a sigmoidal kind of a behavior. Now, the exponential model and the logistic model are based on single species model. So in this case, we are just talking about tumor as a single species. But we have seen that there is a lot of interaction that happens in the tumor microenvironment. So there is a cancer cell, there are immune cell that is coming into picture. There are various molecules that come and interact, and using those molecules, cancer cell kind of try to grow and proliferate, right? So there is a lot of interaction that happens. So therefore, there is a need of what is called interacting models. And there was this model was developed in 1994, and this is one of the pioneering work in cancer modeling using differential equation, where the cancer cell is modeled as a prey-predator type of system, where we can see that the natural killer cell or cytotoxic lymphocytes that is denoted by E here, this can be considered as a predator, and T can be considered, this T is a tumor cell here, this can be considered as a prey. So the cytotoxic lymphocytes, they are preying on the tumor cell, and in absence of the immune cell or the effector cell, cancer cell is assumed to grow logistically. So there is a logistic equation here, and because of the presence of the immune cell here, the some portion of the tumor cell gets eliminated, and therefore there is a minus term here. Tumor cell, they grow logistically, and the term here S is the constant source of immune cell. So they're assuming here that there is a constant fixed source of immune cell in absence of the tumor cell. And this term here denotes the recruitment of the effector cell uh, because of the presence of cancer cell. And this is considered to be of this type, MMT type. MMT type is a response that is commonly used in mathematical models, and G denotes the half saturation constant in this case. So the main result of this work was where they have done parameter estimation of the model parameters, and this they have done using the growth of BCL1 lymphoma in the spleen of mice. So they have used a mice model, and they have injected this cancer in the mice, and then they have studied the growth of this over the period of time. And using that data, they have estimated the parameters of this model. This lymphoma is a type of a cancer in the lymphatic system, which is part of the immune system. And BCL1 is nothing but a oncogene. So that we talked about in the cancer hallmark section. So when this gene BCL1 gets mutated, that causes a cancer, and that cancer is named as BCL1 lymphoma. So in this, what they have done is they have estimated the parameters of the model. This was the main work. And with some numerical simulations, they found that there is a cyclic fluctuations in the number of uh, leukocytes and tumor cells that you can see here. So there is a fluctuations in the population of the immune cells and the cancer cells. And the oscillation period was found to be around 3 or 4 months. So in this case, one of the major observation is that cancer cell fluctuates and they go to a dormant tumor state, where tumor cell do not die, but they remain at a dormant state. And another study they have done is the global stability analysis, where in this case they found that there is a threshold value of the initial tumor population. And if this initial tumor population is lesser than this threshold value, that is 8 times 10 power 7, if the tumor cell is lesser than this value, then they found that tumor will remain dormant, but they do not get cleared, they do not die. And the second result was, if this threshold value was greater than this, if the initial population of tumor cell was greater than this threshold value, then they showed that the tumor can escape the immune regulation, and there is uncontrolled growth of the tumor cell. So this was the major study that was done, and this was done in the year 1994. This paper is published in Nonlinear Dynamic of Immunogenic Tumor. This is the title of the paper, and was published in Bulletin of Mathematical Biology. So this is one of the pioneering starting work in cancer modeling using differential equation, where they considered a compartment-based model for understanding this. Now, another extension of this model was where they took the cytokine called IL-2. So this is again a compartment model. The first two equation remains the same as the before, and there is a new compartment that is added here, that is nothing but the rate of change of IL-2, that is a cytokine. This cytokine IL-2 is the main cytokine that is responsible for lymphocyte activation, growth, and differentiation. So during late 19th century, the main research on cancer was focused on immunotherapy. And in this case, a number of studies were done where they found that IL-2 can be used to boost immune system, and it can help fight the cancer cells. So this was the motivation for them to kind of build up this model. And in this case, the additional thing that they have used is here the new parameter that comes in, the S1 and S2 here. The S1 here, it denotes the adaptive cellular immunotherapy. This is what they call adaptive cellular immunotherapy. That is the immune cell that is injected in the body, and they have considered this as a constant S1. And the second term S2, it denotes the the injection of what is called IL-2, and this scenario they have considered here as a interleukin therapy. So with only S1, they are considering this as a adaptive cellular immunotherapy, and with S2, they consider this as interleukin therapy, and with both of this, when S1 and S2 are greater than 0, they are considering this case as a combination therapy. The parameter C here, it is referred as antigenicity of a tumor, that denotes the ability of the tumor, ability of the immune cell to recognize the tumor cell as a foreign particle or abnormal. So this was the extension of the previous model, and let us come to the results of this study. So in this study, what they have shown is like there is a threshold value of C that denotes the ability of the immune cell to detect the tumor cell. And whenever the value of this C remains between 0 and this value, there is a large tumor size, almost the size of its carrying capacity, and the system is stable. So this means that whenever the antigenicity, that is, whenever tumor cells, the ability of the immune cell to detect the tumor cell is less, the cancer cell grows to a large tumor size, and it almost reaches the carrying capacity, that is the maximum size. The second reason is where they showed that if the value of C is between C0 and C1, where C1 is this value, and as the value of C increases, there is a periodic solution that comes in, that is the half bifurcation that happens with respect to C. And with the value of C increasing, the periodic solution, the oscillations of the periodic solution gets dampened, and the system or the tumor stabilizes around the dormant tumor state. One major observation in this was that tumor free state was never achieved in this case. Like the equilibrium state in this case was, one state was where large tumor size was there, another state is where tumor is at the dormant state. So there is a some tumors population, and it is not 0 in this case. So this figure kind of depicts here. The first one is where there is the tumor cell population reaching the carrying capacity. So there is a stabilization around the carrying capacity. And then as the value of C increases, there is a oscillation that happens, periodic solution that comes in. And this periodic solution dampens, and it stabilizes around the dormant tumor state. Now, this study was without the treatment. Okay, so in this case, what they have done is they have not considered S1 and S2. The another study that they have done is with respect to the treatment, considering S1 and S2. That is adaptive cellular immunotherapy, that is denoted by S1, and second is the interleukin therapy, that is denoted by S2. So in this case, what they have found is in a case 1, that is in the case of adaptive cellular immunotherapy, when S1 is greater than 0. So in this case, they have found that tumor free state is achieved. So tumor free state is achieved, and this is stable. That is, the solution will go to the tumor free state if the value of S1 is greater than this threshold value. And if this is lesser than this threshold value, then the tumor free state becomes unstable. So this is the result that they have shown in this case. Another case is where they have only considered IL-2, that is S1 is 0 in this case, and S2 is positive. And in this case too, they have found that there is again a threshold value of S2. And if the value of S2 is greater than this threshold value, the tumor population is cleared. But in this case, one observation that they make is the tumor cell grow unbounded, and the system will tend to this state, which is not desirable, because if the immune cell is growing unbounded, it may cause a lot of other complications in the body. The third case is where they have considered both S1 and S2, and again they have found some threshold based on which they are saying like tumor-free equilibrium state could be achieved and is stable. So let us quickly kind of do the biological interpretation of this. So whenever there is a low antigenicity, that is when immune cell is not able to detect tumor cell properly. So in this case, it is found that there is a periodic solution. The tumor cell will spend some time at its carrying capacity maximum, and then the remainder of the time at the dormant state. And this behavior could explain the long-term reoccurrence of tumor within the host. Another case is the case with unbounded where there is unbounded growth of the immune cell, and this scenario could explain the side effect that is seen in cancer patient receiving immunotherapy. So this is the biological interpretation. Now coming to another model, this is uh, extension of the previous model where a new compartment is added here. This compartment is where they have used what is called TGF beta. And we have seen that TGF beta has several roles. It has angiogenic property that helps in the growth of cancer cell through formation of new blood vessel that supplies the nutrient. And it also uh, act as a immunosuppressive growth factor. So it also suppresses the immune response. So this behavior of this function of TGF beta is incorporated here. So in this case, previously in the previous model, we did not have this term here. We just had a CT. So this term here, gamma S, it denotes the immunosuppressive property of TGF beta. This is one uh, way in which TGF beta is incorporated here. Another property of TGF beta is the angiogenic property, and that is incorporated in with this parameter. So there is a growth, there is an addition in the growth of the tumor compartment because of angiogenic property of TGF beta. Now this is the result that they have got. So with respect to TGF beta, what they found is P4 is the growth rate of TGF beta, this parameter here. And again, in this case, they have found a threshold value based on which they are saying that if the value of the production of TGF beta is greater than 2.85, the immune system is no longer successful in defending the tumor. And therefore, in this case, tumor mass, uncontrolled tumor mass arises. So this is the observation that they make here. Now they have again extended this model to a treatment-based model where they have kind of used a different expression for the growth of TGF beta in this case. So that they are telling this as a siRNA treatment model, which kind of suppresses TGF beta production by targeting the mRNA that codes for TGF beta. So this is a modification that they make. So in place of this, so they have a new expression here where V is an addition term that denotes the treatment with siRNA. So with siRNA, what they have found is the tumor cell density exhibits sustained oscillation with a low amplitude. So this was the case. This first figure is the case where P4 is greater than 2.85. There is like uncontrolled tumor mass, and the tumor mass reaching the carrying capacity. Whereas with treatment, what they have found is there is a sustained oscillation that happens in the tumor cell population, and this case does not arise. Now, there are some models that are developed using a stochastic differential equation. Ordinary differential equations, they are useful and they give interpretable results, and they are easy to handle unless there is highly nonlinear system. But they also have a disadvantage that they do not capture the uncertainty or the random fluctuation fluctuations in the biological system. And because of this, there is what is called a stochastic differential equations that are developed and studied extensively. And stochastic differential equations, they kind of the use the technique of what is called Ito's formula, and this is a fundamental tool for solving and analyzing stochastic differential equation. The various numerical schemes that are used for handling stochastic differential equations are Euler-Maruyama method, Milston method, stochastic Runge-Kutta method, etc. So apart from the deterministic compartment-based models, these models are extended to stochastic models to capture the uncertainty in the system, cancer system, because cancer system is a complex system involving a lot of random interactions between cells and molecules. So therefore, another approach of uh, handling or understanding cancer dynamics is through development of stochastic model, and this type of modeling is a little more realistic because it kind of captures the biological system better. So in this, the basic idea is if we have stochastic differential equation is represented in general in this form, and there is a operator that is defined differential operator, and if this operator acts on a non-negative function, then we get this expression, and using this we define what is called Ito's formula that is given by dv here. So Ito's formula, it kind of gives a differential of a stochastic process, that is v here, and this v is a function of x, that is the variable of the model, and it kind of helps us to understand the qualitative behavior of the solution of the system using this equation. So now coming to stochastic tumor immune model. So this is again, if you just see this part, so this part was the model that I discussed with IL-2, interleukin 2. And in this model, what they have done is they have added some stochastic parameters, some random perturbations they have done, and then that model is extended to a stochastic model. So in this case, the variable X denote the effector cell that we saw before, Y is the tumor cell, and Z is the cytokine IL-2. So this is the same IL-2 model, 3-compartment model that we have seen. And here the perturbation is introduced in 3 parameters. One is the natural death rate of the effector cell mu2. So mu2 is perturbed, and this is the expression. So we have mu2 plus there is some perturbation that happens here. Similarly, the growth rate of tumor cell R2 is perturbed, and then the natural death rate of IL-2, that is also perturbed. So with this perturbation, so they have kind of developed this stochastic tumor immune model with IL-2. And in this case, this BIs, they are nothing but they are mutually independent standard Brownian motion that captures the stochasticity in these variables. The basic results that they prove is the existence of solution using Lipschitz condition and linear growth condition. So this comes under the well-posedness of any mathematical model. Another interesting result that they have shown in this case that highlights the importance of using stochasticity is they have found that if the intensity of the noise that is introduced in the model, if this is high or if this is greater than this quantity here, then there is a extinction of tumor population. So this is an interesting result that kind of, you know, highlights why stochasticity needs to be introduced in the model. Another result that they show is that if this condition is violated, that is, if the intensity of the noise is less, then there is a persistence of tumor population. So with this observation, these are some plots here with respect to stochastic noise. So the first plot here is where the intensity is 0, that that means there is no perturbation, and there is a there is some population of cancer cell, some population of effector cell, and there is some population of IL-2 interleukin. And the second plot here is where they introduce perturbation, and the intensity they have taken is sigma 1, sigma 3 they have taken 0.1, and sigma 2 they have taken 1.15. And in this case we see that the tumor population goes to extinction, right? There is no tumor population, it becomes zero. There is only effector cell population. And with lower values of the noise intensity, there is a fluctuation in the population count, and this fluctuation is shown to kind of reduce if the intensity is taken higher. So this observation kind of tells us about like why it is important for to introduce noise, and noise has a significant impact on the behavior of cell population, from extinction to persistence. This fluctuations in the system due to various factors, various environmental factors, or various random interaction between the cancer cell and immune cell. This becomes significant, and this behavior can be captured using stochastic differential equations. Now, coming to the second part of my talk, here I would like to kind of briefly talk about uh, mathematical model, the machine learning models in cancer prediction. And this I'm going to talk about uh, in the context of breast cancer detection work that we have done. So with the vast amount of the data that we have currently, like various mathematical models have been used to understand and identify patterns and relationships between the data set. And in case of oncology, machine learning and deep learning models are used in various fields. Some of them are risk assessment, where you want to identify uh, people having higher risk of getting cancer. It is also used in early detection and diagnosis, prognosis, and survival predictions, where you want to predict the survival chances of a patient having cancer, the selection of treatment. So these are various areas in which machine learning models have been used currently. Now, before kind of going into the results of our study, I would like to kind of briefly talk about the breast cancer scenario in India. So if you see Indian context, breast cancer is one of the most common cancer in India, and it accounts for almost 27% of all cancers in Indian women. It is also found that every 1 in 28 women, they are likely to develop breast cancer during their lifetime in India. And if you see the estimated cancer incidence, estimated cancer cases in India in the year 2022 was around 14 lakh 61,000, and it is estimated to increase by 12.8% in 2025. So this is one study that was done where they have estimated the where they have done the forecasting, and they're saying that this may increase by around 12.8%. So cancer is cancer incidence is increasing at an exponential rate, and the most common is the breast cancer in case of humans. Now let us quickly see why it is important for early detection of cancer, and what is the corresponding rates of survival. Now, the fundamental question is like, if somebody gets cancer, can it be cured? So this can be get cured, cancer gets cured, but if it is detected at the early stage. So if cancer is detected at stage 1 or stage 2, the survival chances of the patient exceeds 90% for cancer, including breast cancer and colorectal cancers. And this figure kind of gives an idea about, if cancer is detected early, then the survival chances are above 90% for cervical breast cancer, and other skin cancer. And if it is detected late, stage 3 or stage 4, then survival chances greatly reduces. So therefore, it becomes extremely important, for early cancer detection. And this early cancer detection allows for more effective treatment options and a better chance of, curing the disease. So in these scenarios, machine learning models can be employed with a huge amount of data that is available. Machine learning models greatly helps in detecting cancer at the early stage. Now, if we see the normal detection procedure in case of breast cancer, this is normally done through what is called a triple assessment, where they go for a clinical examination, the imaging technique, the pathology, where they do biopsies and fine needle aspiration. And with all of this, they kind of combining all of this assessment, they do the confident diagnosis in 95% of the cancer cases. Though this triple assessment, it is a thorough and reliable procedure for detection, but it also has several disadvantages. The first being, like, this involves a lot of time, right? There are a lot of things that is happening, so it is time consuming, it is also expensive, and the biopsy that is being done, it is invasive, it can be really painful for the patient. So the question now here is, can we kind of make this process faster? Can we kind of expedite this process so that there is cost effectiveness comes in? And in this case, like, machine learning models are being used so that this process is done faster and cost effective, in a cost effective way. Now, the what that we have done here is, like, we are given a set of or breast cancer data set that consists of 569 samples, and this has 32 attributes. And these attributes, they are kind of extracted from a digitalized image of fine needle aspiration of breast mass. And this problem is basically a classification problem, where we want to kind of given a relevant uh, data of a breast cancer patient, can we say that patient is having cancer or not? In this case, it is whether the cancer is malignant or benign. So in this case, this is uh, some of the features of the data set that we used, that I have shown here. And in this case, we have a column called diagnosis that has two classes, malignant and benign. And our problem here is to kind of classify the cancer cases to malignant or benign. So in this case, we have done basic data pre-processing. And we have used a logistic regression model that is commonly used for classification problem. And decision, we have used decision tree. So we have done a comparative analysis of this model. The third model that we have used is random forest. So these are the three models that we have used. And we also wanted to kind of to get a comparative study. We have also used what is called a feed forward neural network model for the same data set. Though our data set is just 569 cross 32, it is a small data set, but to get an understanding of its the performance of neural network and kind of do a comparative study, we have also used the neural network model in this case. So I think I don't have much time, so I'll directly go and show the result of this study. So these are various evaluation metrics that we have used here. We have used accuracy, precision, sensitivity, specificity, and based on this metric, we have evaluated the performance of the models that we have used. So this is how our results look like in this case. So logistic regression, decision tree, random forest, and neural network. And among the machine learning models, we find that random forest does a pretty good job in terms of accuracy, sensitivity, in terms of all the four metrics. And above all, compared to all the three machine learning model that we have used, the neural network is doing pretty good in this case. So, in this case, so we have shown that neural network model is good, and this is the values of the hyperparameters here. We have used 200 epoch, just to hidden layer of the neural network with learning rate as 0.001 and activation function as ReLU and Adam optimizer. So in this case, like because our data size is small, we may not it may not sound good to kind of generalize this result, but we are kind of in a process of getting more data from the tie-up centers that we have here, and we are trying to test those cases using this model that we have developed. So we also did a comparison with one work that was done, and this work they have used the same data set, and they have used RNN and LSTM model, and here they have reported accuracy of around 85% and F1 score of around 86%. So in comparison to this model, our neural network model is doing pretty good in terms of accuracy and other metrics. Now coming to the current work that I'm doing, I'll take just 2-3 minutes to discuss this. So we saw that cancer is nothing but it is a multi-scale process. So for understanding cancer, a thorough understanding of cancer disease requires integration of various models across different time scales, from molecular scale to cellular models. All of this, if there is an integration of this model, that can give us a better idea, better perspective of understanding the disease. So this is one work that we are currently doing. The second work is tumor volume prediction and forecasting with a time series data using RNN and LSTM model. So this is another interesting problem where we want to kind of forecast tumor volume given the time series data or sequential data of tumor volume. The third work that is currently being done is prediction and survival analysis in glioma cases using machine learning and deep learning models using the MRI data. So this is what I had to kind of present in today's talk. I think I have taken a lot more time than what was given. So this is the references that I have used. So thank you all for your listening. Saira.
Sairam, everybody? Am I audible?
Yeah, I think you are audible. I can hear you.
Okay, so if anybody has any questions to ask to
Hi, Bishal. This is Bishal from your department.
Oh, hi.
So can you please go to this slide where you mentioned the data of several machine learning models, where you chose one over another, you showed that this gives the best prediction.
Yeah, I'll just show that. This is what you're saying?
No, the previous one where you showed yes. The next, no, where you showed the
data.
all the models.
This one.
Yes. After that.
Before this, I think. Yeah.
No, not this slide where you showed like
the output of all models,
Okay.
random forest, regression model.
Okay, I think this one.
Yes, this slide. So I'm just curious to know, how did you define that, for example, a random forest having this 0.89, and maybe if this is in percentage, then 89%, it is more accurate uh, and predicts the result better than like logistic decision and neural network models. So given that, like if we talk about accuracy, it is like 89. If it is percentage, 89%. Okay, but neural network has had this 92% accuracy, but it had a little bit more sensitivity. But then if we switch to decision tree model, then it has just 0.03 percent differs with like 0.03 from random forest, while yes, 0.02 in sensitivity. So how do you say that this random forest, it gives the better prediction? Why not like decision tree or like neural network?
So yeah, so random forest, I'm saying like in case of like, because we evaluate the performance of a model across all these metrics, like accuracy only may not be sufficient in saying this model is good, because that also depends on the model that we are using, the data set that basically that we have, because there may be like imbalance in the data set. So because of this things, like it is the model is evaluated across all these parameters, and based on this, the effective model is kind of chosen. So when we were kind of comparing the models, machine learning models, I'm just talking about the first three here. So in this case, like we find that if you see individual kind of a metric here, right, then in this case, like random forest accuracy is kind of pretty higher than, like comparatively higher than the other two models. Similarly, like the sensitivity, precision, and the F1 score. So based on this, this is what I am saying. But uh, one thing, the kind of limitation to our work is the data size that we are using. It is kind of size is less. So based on the given data set, this is what our models are predicting.
Okay, so that I understood, but I wanted to understand, like there is only 0.03% difference between decision tree and random forest in accuracy, and 0.02 difference in
Yeah.
sensitivity, right? If we
Yes.
consider these two parameters, then how does a random forest, how does this 0.03 or 0.03 make a difference? uh, that using this difference you consider random like forest better than decision tree.
So in this case, I think if you are just looking at this, then we can say that both of this model is performing like almost the same. If you are just looking at accuracy and sensitivity, if you are looking at that. But if you see the precision, then there is a difference that we can observe. There is a pretty much difference.
Yeah, but the logistic regression also have almost same precision as random forest, right? 0.82 and
Yes.
0.84. So there is only 0.02 difference in precision, difference in precision and sensitivity, and 0.04 difference in accuracy. So can't we like say then that logistic regression also performs like as par with random
Yeah, I
forest?
think. So
Okay, so
yeah, I
another,
think yeah, another interpretation can be that like logistic regression is also doing as good as random forest for this
as
particular study.
all right. So I another point, I didn't understand what this accuracy means. Like random forest, so this score is
So
like
accuracy. So
0.89.
actually,
So
I have defined here actually. So I think I skipped this slide. So this, in case of classification model, so this metric is defined in this way. Like if you just see the accuracy. So in our data set, what we have is like we want to classify a cancer as benign or malignant. So it is either 1 or 0 in our case. So this is the two classes that we have. And
Okay.
accuracy is defined as the percentage of prediction that are correct, like the true positive cases predicted positive and it is actually true. The true negative cases, it is 0 and model is predicting 0. So it is like among all the predictions, how many are kind of accurately predicted, that kind of gives the accuracy of the model. And similarly, other parameters are defined.
So that means you already know what your data is, right?
Yeah, we already know. In case of this study, we already know the data because that data only we have used for doing the study. So in this case, we had a data size of 569 cross 32. So this is how it looks. I have just put a screenshot of that. So we have a column in the data set, the name is diagnosis. And here in this case, we have M and B cases. And we have the other features here. So based on this data set, we are training our machine learning model. And now given a new input to the model, it will be kind of saying me whether it falls in which category, whether it is malignant or benign. So this is the problem that we have done here.
All right. All right. Thank you. Thank you very much.